vast majority of rare diseases are actually these single gene disorders. So if we can identify what has gone wrong, we can identify the target, then it just really gives us a lot of potential to try to target the disease right at its very basis, its molecular etiology. This also has a lot of implications for rare disease diagnosis because, again, this is increasingly how we're identifying what the disease is. It's based on this underlying genetic problem. So increasingly, diseases are being diagnosed, characterized by their genetic underpinnings. So I've been working in rare diseases for more than 20 years, and it really felt for a long time like there was very little that we could do for people. And that has just changed radically in the past, especially the past 10 years or even 5 years. The rate of change, our understanding of these diseases, the science that is available right now to treat these diseases, the strategies. I think the commitment of the scientific, the medical, and the patient communities to now very deliberately and very focused way developing some of these very targeted precision and even in some cases personalized therapy. It's just incredibly exciting. It just feels like now we just have such incredible opportunity to really make a big difference in people's lives. If we're able to very specifically define what the disease is, it opens up the possibility of personalized or these precision or targeted therapies that we've been talking about with some kind of a genetic medicine. So that's the first very important step. So a lot of our diseases in the era before, really, we understood much more of the genome, and the genome was sequenced, and now we have a much greater ability to sequence patients rapidly and try to really hone in on what the specific defects is. Before that, a lot of these diseases were under syndromes. They were described clinically by their characteristics, and as we're finding out now, even diseases that may look a lot alike clinically may have very different underpinnings. So of course that has enormous implications for how we would treat somebody. The better we can understand that, the better we have a chance to intervene, and to intervene more specifically, that hopefully is going to produce a more effective and safe treatment for that patient. But also, for many of these diseases, they are chronic and they are progressive. So we would like to intervene certainly before patients have experienced irreversible consequences of their diseases. And for some diseases, particularly neurologic diseases, there may be windows of opportunity where we have to intervene to make a difference in someone's life.
The good thing about that is, in contrast to other diseases, we know exactly what the problem is, and we know in principle how to fix it. And in the case of monogenic disorders, the problem is that the patients have 2 copies of a defective gene, and so certain cells in their body don't make a particular protein, and that causes the disease. And I was working with diseases that are so rare that nobody in their right mind would probably develop a therapy for them by themselves. If we could develop this into a platform to make it easier for the families of patients with diseases like the ones I was studying, I think that would be, I realized that would be just a really wonderful thing to do, and I'm thrilled to be involved in many projects that get at that. It's something I never anticipated to do when I started running a research lab, but it's actually even better than running a research lab. Gene therapy offers a way to treat those by essentially putting in a working copy of that defective gene into the patient's cells. You can do that in kind of a platform way, which is to say you could use the same delivery system to deliver different therapeutic genes to different patients with different diseases. But it's kind of like a therapeutic platform, and I think that to me is the excitement of this, because in principle this general therapeutic platform is applicable to a large fraction of single gene disorders, which is a large fraction of rare diseases. When we talk about gene therapy, at least in the sense that we most commonly use the word, it refers to putting back a working copy of a patient's gene into patient cells. So we'll be talking about monogenic diseases where the patients have no working copy of a particular gene. And the gene therapy then is to put a working copy of that defective gene into the particular cell types in the patient to correct the disease. And if you can do that in enough cells at the right time of development and the right time in disease progression, you could cure or prevent genetic diseases. To move a gene therapy from a lab to a patient takes an awful lot of time and money at the present time. And I think a lot of the programs we're working on is really designed to sort of shorten that pathway. But at the present time, it takes many years, it takes millions of dollars, and a lot of regulatory steps.
The gene therapies, right now they are very expensive to manufacture. They're very hands-on. They have multiple steps, small batches at a time, and the cost of development, the assays, and characterizing these products. It's very hard to do. There aren't a whole lot of people that know how to do them. We don't have enough capacity, and the cost is just very, very high. So this is an area of just active development across NIH, but also in industry, to try to be more efficient about that process, so we can make more product, we can make it better, we can make it faster, and we can do this for many more patients.